Trying to get it to log in.
Hello, everybody. We're going to just be starting in about 30 seconds to 45 seconds. We're just trying to get our YouTube feed up and running. Just give us 1 second, and we'll momentarily be starting our show tonight.
What's up? I'm trying to get the YouTube to work right now and it's not cooperating. Yeah, I don't know what to do. It works on PC.
It was working before.
No, we didn't authenticate it.
Oh, no.
Alright, Dave,
All right.
do an intro.
All right, guys. Today we're going to be talking about the three-parent child born in the United Kingdom, basically by in vitro fertilization methods. Today we're going to be talking about mitochondrial DNA versus nuclear DNA, what the difference between the two is. Going to go into a little bit of detail about the structure of mitochondria, and we're going to also go into a little bit of detail about what ATP is and how it affects development. After that, we're going to go into the actual IVF, the in vitro fertilization process that was used for this particular child. And we'll talk about the ethical dilemmas assigned to this particular procedure. So, as soon as we get... All right, we're going to have to just do the Facebook Live tonight, which is okay, no big deal. We'll get this situated.
All right, cool.
So, sorry for the technical difficulties. We'll be up and running on YouTube next week. Sorry about that. We tried to use a Mac PC, and neither of me or Nate are Mac guys, so it's quite okay. All right, so starting off, we're going to talk about what nuclear versus mitochondrial DNA is. Everybody knows that, you know...
No, not everybody, Dave. Start real basic here.
Yeah, so good point, Nate. Basically, what nuclear DNA is the DNA that you get from mom and dad that come together in your zygote, which is the first stage of sperm meets egg, and that's your first cell that will eventually become a full-blown baby, full-grown baby, person. So, what is mitochondrial DNA? So everybody, has a general idea that, genes mean proteins, or genes mean what you're going to be if you're blonde hair or blue eyes. So when I say mitochondrial DNA, most people go, what is that? So the mitochondria is the powerhouse of the cell, and basically where ATP is made. And ATP is a particular molecule that has 3 phosphate groups, which is phosphorus and oxygen.
And ATP is basically like the fuel for everything that we're doing, right?
Think about it as the gasoline to the cell. So when you want to contract your arm and make big bicep, this is my meatheady bicep, you break ATP. So you'll actually break these bonds right here, and it's transferred to a different molecule to do the function of the cell. So it's basically like transferring the gas from your gas tank to your engine to make the engine go, move the tires.
Sure, that makes sense.
So that's pretty much the most basic way I can describe it. And how the ATP is formed is not in the ground where dinosaurs die and then eventually become gas or oil. It's made in the mitochondria. And in particular, the theory goes is that when we were just all one little cells floating around in the big primordial goo, is that bigger cells that started compartmentalizing themselves, like say, how we have a gut, individualized cells have guts that break down little metabolites and substrates to make things that they need, like glucose into energy and different things like that, amino acids into proteins. So what the bigger cell did was it goes nom 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 and ate the little mitochondrial cell and became a 1-part cell. So the big cell would go and eat all the glucose and all the sugars around and deliver that to the smaller little mitochondrial. And in essence, this little guy in the middle has its own set of genomes, a set of genes to make proteins that does the job that it needs to do. And in turn, being fed from the bigger guy, it gave energy to the larger guy. So It's a nice little symbiotic relationship.
Sure. So what gave energy to what, Dave?
So the
Break
little
that
guy,
down.
gave energy to the big guy, and the big guy gave all the things necessary to make the energy to the little guy. So you know how you see those little sharks swimming around? You see little sharks, big sharks swimming around, but they have those little fish that swim in their, their wake and eat all the scraps that the shark leaves behind.
Yeah.
And then the little fish will eat all the little parasites off the shark. Kind of like a, hey, you help me, I'll help you type of relationship.
Host-parasite relationship.
Exactly. So that's how we have a nuclear genome that has genes that decide whether we have arms, legs, antenna, or if we're a fly, we got wings and we can flap around.
Mm-hmm.
That'd be pretty cool if we had wings.
That would be pretty cool.
Pretty cool, right?
Get down with that.
So the mitochondria in our cells will produce the energy, and vice versa, the cell will interact with it in a symbiotic type relationship. So what happens if the mitochondria are defective is that the kid or person that's afflicted with mitochondrial disease won't actually develop to full adulthood and will actually die in the earlier stages of development. So
this is kind of prevalent, right? Like it's not that rare?
No, it's actually very prevalent. It's actually 1 out of 40,000 live births will have a type of mitochondrial disease. So if you think about it this way, there's 3.9 million new births every year. And if 40,000 out of every 40,000 there's one mitochondrial disease, that's a big number. As Nate thumbs through our stuff.
Thought you wrote it down somewhere, but yeah, that makes sense.
Yeah,
All right.
it's actually on the 3rd page. But moving forward, you don't get mitochondria from both dad and mom. So when the egg gets infertilized by the sperm, the little guy swims up and goes inside. I think we've all had sex ed where we know the little sperm, he swims and goes, okay, I'm here, I win. All the other sperm lose. The mitochondria that's in the sperm from the dad actually gets degraded and disintegrated. So it's actually not used in the child. So you have your mom's mitochondria, I have my mom's mitochondria, and we have our mom's mitochondria. So there's actually a genetic Eve that's termed genetic Eve as our first mother of all our mitochondria. So that's actually a pretty cool thing.
Yeah, I didn't
might
know
talk
that.
about that in another topic of maybe human evolution. So stay tuned for that. Moving forward, if your mom has defective mitochondria, or if she gives you maybe 3/4 good mitochondria, 1/4 bad mitochondria, you'll be able to live and be able to function as a normal individual with a regular, what's called phenotype, the thing that you actually observe from based upon the genes or the genetics underlying the phenotype. In particular, we're going to be talking about Lee syndrome. And what Lee syndrome is that a higher percentage, greater than 50% of the mitochondria that you get from your mom are defective. And it's no fault of her own. It's just that when gametogenesis, the making of the eggs in your mother, actually occurs, there's something that goes a little haywire, whether it's, age-related, whether it's, just it's in your background from her mom's mom.
Sure.
And, there might be a tipping point. So there's actually a fair degree of randomness to this, where some cells of the daughter, when it separates off, maybe you get 50% really good and 50% really bad. Probably not really that great for your offspring, but there's also cells that actually get 20% good and 80% bad.
Sure, and this is also pretty common in older age-related infertility, right?
Exactly, and there's a process called body bars and different things like that where, or polar bodies, sorry, excuse me, body bars are completely different. That's X-linked X chromosome mosaicism. Basically, it comes down to the fact that when you get older, your eggs are in hibernation, and as you keep going and going and later for reproductive years, as our society is now doing, those eggs get a little defective. It's kind of like sitting a battery up on a shelf for 10 years.
Sure,
and you go,
eventually it's just going to go bad.
Exactly.
Right.
It's just
Use
like
it or lose it, baby.
Exactly. So, consequences of this is that you might have defects in your mitochondrial DNA, or the mitochondria just break down and they just don't separate properly, and you get what we're going to actually go into is the
so
details
this
about it.
This was pretty recent, Dave, right? Like 2014, I think?
Yeah, and actually the first time it was actually performed was in 2009 under with rhesus monkeys actually in Oregon.
Sure.
So in particular, there's 3 methods that this in vitro fertilization was theorized back then to actually work. In particular, we're going to be talking about spindle transfer. And basically with spindle transfer is that, actually I'm getting a little ahead of myself, is we're going to take out the nuclear DNA from the mom's egg and put it into a donor egg that has good mitochondria. We'll get into a little bit more detail about that. But let's talk about the mitochondria in general and what this Lee syndrome actually does.
Definitely.
And
so Lee syndrome is a type of mitochondrial disease, right?
Exactly.
Okay.
And usually 80% of the time, it's nuclear DNA that's the issue. And in our particular case, what we're talking about today is 20% of the time is mitochondrial DNA defects. And that could be caused by any number of things, whether it's reactive oxidative species, whether it's just DNA damage, whether it's just defective mitochondria, just in general, where it just doesn't form properly, different things like that. So I'm going to go over just a brief little snapshot of what the mitochondria is in terms of structure and what the affected portion is in Lee syndrome.
Cool.
So you have complex 1, you have complex 3, and complex 4, and then you have this nice little quinone pool, which actually holds electrons and helps transfer from the inside to the outside H protons. And what the H protons going from the inner portion of the mitochondria, because there's inner, matrix, membrane, cristae, membrane, the rest of the cell.
Sure, so just like a layering.
Yeah, and basically what it does is you want to put all those H's up here to cause what's called an electrochemical gradient. And that energy stored in that gradient is used to make ATP. So what you do is you send an electron up through what's called complex 1 or NADH dehydrogenase. And what that does, it pumps out 4 units of H protons and transfers the electron over into the quinone pool. The quinone pool goes to complex 3. Complex 3 goes to cytochrome C. Cytochrome C dumps it off into complex 4, which is cytochrome C oxidase. And that's where O2 will catch all the electrons and H's and make water and power ATP synthase to make ATP.
Okay, so you pretty much described the process that it would normally take for us.
Exactly.
Gotcha.
But in Lee syndrome, what they noticed was that the first complex and quinone pool is the effective portion. So you can't even get to this portion of it where you actually make the energy, and it's already defective over here. There's some transfer of electrons and some protons going through, but think of it like this. You're stepping on the gas, but for whatever reason, there's a little kink in the line, and it can't get the gasoline from your gas tank to your engine. So you have a little gas spilling out here, you have a little gas spilling out here.
Leaking the hose.
Yeah, leaking the hose. So that's basically what happens with the affected portion of this. So what they decided to do was to say, you know what? Let's get rid of all the bad mitochondria. Let's just say, let's not even go through, try to fix the mitochondria because it's really, really, really impossible to do. Because at that point in time, there's so much interaction between the nuclear DNA and the mitochondrial DNA that it really doesn't make sense to go that avenue. It's too expensive.
Sure.
We won't have the resources. We don't have the technology to do that as of yet. So what they decided to do to solve this problem, if you can just give me 1 second to erase our little picture right here, is to take, I'm going to use black to signify the mother egg. Okay, so this is mom. Okay, and we're going to use red to signify donor egg. Okay, so this is donor. So in the case of the donor of the case that we were looking at, it's from a portion of the United Kingdom around Newcastle University. So the individual in question actually comes from a population very similar to the mother's population. So you don't want to use somebody, say, from, in the studies that were done, a different species of mouse to a different species of mouse.
Sure.
That are separated by millions of years of evolution. So we want to use something very homogeneous.
Does this have to be someone of the same blood type?
No, it actually doesn't matter. I actually looked into that, yeah.
Interesting, okay.
So we're going to signify good mitochondria with these guys. These nice, big, bright, okay, just like that. And how about we signify the nucleus with just, and we'll put
larger circle.
a big larger circle and we'll put like an X through it, okay? Just to signify the difference. And then up top, we'll use this color to signify bad mitochondria that are just defective. And I won't even give them any type of shape. So basically what they do is they take the mother's nucleus, okay, that's the mom's nucleus, which is bueno, it's good, has all the genes that you need. And basically they take it out. Let me use a red guy. So they use a nice little needle, okay, and the guy's all in hazmat gear basically. He's got a full suit on, can't get any of his DNA in there, otherwise it'll screw the whole thing up, or she, sorry. But basically, you have the mom's nucleus come out.
Mm-hmm.
You put it into the little tube here. Okay. And then you take the same needle and you take the nucleus out. Okay. And we just chuck that away. Okay. We just throw it all away. So let's get rid of this. It's thrown away. Okay. Okay. And we take the mom's nucleus that we just extracted from up here, and we insert it directly into the, let's encircle it in black,
Sure.
so we can signify nucleus right there.
Mom's nucleus and the donor's egg right
Yep.
now.
So at this point in time, they take nice little, let's use this. Talking about that little spermie with the little tail right there, you know. So he's going to go in at this point and fertilize the egg. So this is where the concept of 3 parents has come from. 3 parents. So 3 parents, just to make it easier for the general public to understand what's actually going on. So at this point in time, this egg will be put into a petri dish and it'll grow into a 16-cell blastocyst. And at that point in time, they'll test for viability. Okay. And at this time, this whole process is done in not just 1 time, 2 times, 3 times, 4 times, actually done in 5 to 6 times to actually make sure that they get a viable blastocyst. And they'll take the 3 best blastocysts and insert it into the mother.
Gotcha. Okay.
So the original mom, who doesn't have this egg,
Mm-hmm.
with the bad mitochondria that's taken away, and this will divide and 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 divide into a sphere, and they'll actually implant that into the mother. So, and at that point in time, there'll be 3 of those in the mom, and they'll have hormone therapy, this type of therapy, that type of therapy. They'll keep levels of progesterone low, they'll keep this high, they'll keep human gonadotropin high, and basically the fertile egg will grow and grow and grow. And as some people may know, with IVF, there's a high chance of twins because of this multiple-pronged approach where they don't just take 1 fertilized blastocyst to put it in there, just increases chances of having children.
Theory too.
So if you're 35, 36, you know, even up to 44, I know Janet Jackson just got pregnant through IVF at age 50. So,
really?
Yeah.
Wow.
Yeah, there's a little fun fact for you. They use multiple types of blastocysts to try to get the woman pregnant. Because at that point in time, who's going to complain about having twins? You know, you wanted kids, you wanted kids, you wanted kids.
In general, sure.
Exactly. So it's very common to have twins and triplets, actually, as well.
This sounds pretty cool, man.
Yeah, so that's actually just the basic science behind it. You know, it took, I think, about, from start to finish, working in mice to rhesus monkeys to now humans, it's taken about 10 years to get from stage 1 to stage where we're at now. We're in human actual trials. But this all has only occurred in the United Kingdom, and the United Kingdom just recently changed the law in the previous year, in 2015, so that this actually can happen. And this actually leads up to a couple things that I wanted to discuss in terms of ethical dilemmas and what this actual can mean in terms of maybe a slippery slope theory. So basically, say Nate has bone cancer, okay? I never want to wish that, knock on some wood, don't want to wish that on you.
Hope not.
But let's say Nate has bone cancer, and this is Nate. He's like, oh shit, I got bone cancer, oh no. So basically what you want to do is you'll take his bone marrow out, big needle, ouch, ouch,
Yeah.
okay? You'll take it out, you'll fix the problem, or you'll get a donor, that'll be me, and then I'll take mine and put it into you, okay? And then there's particular things, especially with treatment of like HIV, bringing it back full circle to last week, where the gentleman in Germany actually got a donor bone marrow, and it was immune to the HIV virus.
Sure.
Or there's also different types of genetic treatments where you'll take the bone marrow out, put it into a petri dish, and you basically manipulate it with vectors, adenovirus vectors, and basically get good DNA in, the bad DNA out, and you put it back into the person. Now, there's very little ethical dilemma with that, because when you have kids, let's give you a nice little girlfriend, okay, maybe Steph, okay, so these two have kids, and the subsequent babies may have the same condition that you had, so this process will have to happen all over again.
Sure.
So it's a one-stop shop, basically, or one-time fix, but your progeny, or your children and your subsequent generations of children, will still have this type of problem. But with this, in terms of the nuclear mitochondrial...
The spindle transfer, right?
The spindle transfer, what we were just discussing, if you fix the mom, we'll keep the mom blue, and you have dad come in, okay? You mate these two, and you have kids. Theoretically, these kids are all fixed.
Sure.
Yep. So you have all the kids, they're good, they're healthy, and let's circle little Jane and little, I don't know, what are you going to name your kids?
Heck, I don't know.
All right, well, we're going to name it Jane and Jamie, okay, girls. So with the boy, if you get this fix, it ends with him. Like we had said, mitochondrial DNA, mitochondria only come from the mom. So if he has kids, it'll come from his wife.
Sure.
Okay? But these two, in theory, can have kids, 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 kids. And let's say all these kids are girls, and times 2, and it goes down and down and down. It has a multiple effect.
Sure.
So basically,
just stop at one generational line.
Exactly. So you're fixing not just your generation, you're fixing your progeny forever and ever and ever, and so on and so forth. And there's very little study done onto the effects of that in nature, in terms of human society, and in terms of even just mice and in general. There's several hundred generations of mice and Drosophila fruit flies that can go through this process, and we've studied them, but we don't know, exactly how this is going to affect the natural world. So that's kind of one of the ethical dilemmas
Sure.
that face this. And my belief is that if you can cure your entire family of a disease and to never make them actually have it ever again, why would you not do it?
Yeah. I mean, it certainly has all the positive benefits, except for the risk of complications during the procedure itself, which there's always going to be a risk no matter what procedure you're doing. But I guess the one point that you wanted to bring up was the fact that the child itself wouldn't have a choice as to whether this would be done on them, right?
Exactly. And that's another ethical dilemma, where human research subjects are always told, hey, you know, this is the procedure that we're going to do on you, this is the risk, this is the benefit, and basically we're going to get, you know, more information from giving you this drug or this treatment or different things like that. Do you consent? And that's the big thing. The parents may want to consent to it, but what happens if this procedure doesn't work? And then at 10, 15, 20, let's even say 60, 70, 80, that this child has repercussions from this treatment. Can you in full good-hearted say that that child might not want this procedure done at that point in time when they're suffering? So that's the other flip side of it.
Yeah, but then you also have to take into consideration that there's a good chance that kid might not even be there without this type of procedure.
Exactly.
So it's kind of, you know, do you want to take the chance of having a kid that may live a full, healthy life, or, you know, not even have the chance to have a child at all?
Exactly. And
yeah, it's a tough spot.
That's the flip side of it. And I really think that if the clinical trials in rhesus monkeys and mice have been clearly shown to work very, very well. Rhesus monkeys, actually, I have a couple stats. Sorry, guys, I don't know where I put my notes. Oh no, I found them!
You got
Yay! Okay. IVF normally has a 75% chance of working, and with the IVF spindle transfer, 57% of them were viable, the blastocysts.
Sure.
So that's actually pretty good. And that's not including the child actually growing and different things like that. The rhesus monkeys that were actually born from this particular treatment are all healthy, they're growing, they actually meet their cohorts. So let's say, a child at 1 year meets its other 1 year standards. So it's this big, it's this wide, it's this fat, it has good, movement.
You wouldn't be able to tell the difference between...
Exactly. If we were standing in a cage full of rhesus monkeys, God save us if we were, but you wouldn't be able to tell which one was the one from IVF or the one that was actually born normally, through regular copulation.
Yeah, well, I guess we'll just pay attention to this next couple of years and even decades, really, right?
And actually,
especially something like this.
The rhesus monkeys that were first performed with the IVF situation are now coming up to reproductive age. So they're actually producing children, and they've actually produced litters that are viable.
Cool.
So those babies are around 1 to 2 years old now, and they're actually perfectly healthy. They don't show any signs of the mitochondrial disease. So that goes into question, hey, does it actually work, fully?
We got a question here,
Think
Dave.
about whether the child has the right to be told they were made through IVF. The right to know made. I think that also goes into the ethical conundrum, but normal IVF situations, would you tell your child that you normally had been going through IVF or fertilization treatments and different things like that? I think that comes down to the parents, mostly.
Whatever your personal parenting style is, or religious side of it, or...
Maybe we can use the analogy of would you tell your kids that you adopted them?
Probably, yeah. I would feel like that would probably be a good thing to do.
If I adopted a, African child or, a Hispanic child, quickly, I think they're going to know pretty quickly because I'm pretty white, so I don't know. But that's just, me. I would tell my child and be upfront and be honest. Because at a certain point, they're going to know what lying is. Yep. And I don't want to be the one to teach them what lying
every kid
is.
figures out when Santa's not real, right?
Exactly.
So there's a good chance you probably want to have that conversation with the kid sooner rather than later, right?
Exactly. Yeah. That's kind of my take on the matter. All right.
Cool.
If anybody has any questions, please feel free to throw them up, and we'll try to answer them as best as possible. I'm going to take it back just a little bit for us right now.
Yeah.
So, Nate, it's your topic now.
All right, cool. So, the one thing that I wanted to talk about today was I sent Dave this podcast about this engineer who worked at Twitter, Apple, and Google.
Leslie.
His name was Leslie Miley. Yeah, he's a black guy. And one of the things that he found was very difficult about working in pretty much any Silicon Valley company was how not diverse they are. And, you don't really have to spend that much time doing your own research. You can look at any of the Apple reports where they talk about how they want to improve diversity every year. You can look at the same thing that Google says. They say they're not where they want to be, but they're always putting out press releases like, oh, we're working on all these initiatives, blah, 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 blah.
Do you remember the percentage of African American or people of minority backgrounds, their percentage of the company?
Specifically, African American is definitely less than 5.
I think
but then,
around 6
when it comes to like people of Indian descent or of Asian descent, I think it's a little somewhere around there too. I don't know. I don't have that off the top of my head, but I know specifically for African Americans it's less than 5%. And even in leadership, he was the only one.
Oh yeah.
So
do you? I specifically remember, was diversity just about race, or was it other factors?
No. So I think the best way that you can basically think about this is in that podcast, there was a great example of where people keep their ketchup, right? Yeah. I think this is a really stupid example, but we'll go over the same thing. So, Dave, where do you keep your ketchup?
I keep it in the fridge.
Yeah, me too, right?
Yeah.
So, like, if you ran out of ketchup, you'd probably use, like, what, barbecue sauce or something like that?
Sauce.
Yeah, so another similar product that's in the same proximity of what you're trying to grab.
Mm-hmm.
But according to people who live in the Deep South and in England, they keep their ketchup in the cupboard. So if you don't have if you run out of ketchup, you might be more likely to grab like some type of sauce or some type of tomato paste or something like that.
Interesting.
Right? I don't know. But the point is, if you're a business and if you want to make money and if you want to know the type of people that you're selling to, you need to know these things, right? Someone who didn't live in the Deep South or someone who didn't live in England probably wouldn't know that that's where people keep their ketchup.
Yeah, I certainly didn't
Yeah,
until I
and
started the podcast.
they wouldn't have got a chance to try to sell to that person, right? So it's about paying attention to not only where the differences in the people you're targeting are, but just general demographics too.
Interesting.
Yeah, I mean, you only have to pay attention to the last couple of United States Census bureaus. You know, white people are still having babies, but it's declining, and you know, Hispanics and African Americans are growing every census. Yes. Yeah, so I mean, if you want to
so we're going to be a minority majority
at some
country
point.
soon.
Yeah,
I believe they think they said around 2050. I think that's
what everything that they told us in school is. Yeah, I don't know.
Yeah.
I don't have the latest numbers on that.
Yeah, well, I'm all about the what's the browning of America or whatever the hell the right wing wackos call it.
Yeah,
but,
we'll all look the same, and you know, hopefully everyone will know.
Maybe it'll be like South Park, where we're all going with the futuristic people.
It might be, but
diversity, I remember them saying that it doesn't necessarily mean about race or anything like that. I remember them specifically saying, you know, if you're educated at a particular university, and, you know, let's say in Silicon Valley, for example, Stanford's right down the road. Mm-hmm. You know, you're going to want to pick, that's a good school. But I remember them specifically saying that you don't want to just have 90% of your pool, like your engineers, from that particular school.
Exactly.
Maybe you pull from UC Berkeley, or maybe University of Texas, or University of Pennsylvania, or Penn State Temple. You know.
Well, I forget there was another professor that was on the podcast as well, and basically they were doing smaller sample sizes of homogeneous groups of people who had the same background. So maybe they all had the same type of Ivy League background, or maybe they all had the same type of IT background or marketing background or whatever it would be, against a group of people that was mixed. So maybe it was people who went to some Ivy League and some went to community college, or some had masters and some had undergraduate, or whatever the mix of the groups were. Chances are, in these smaller groups, the groups that were mixed outperformed the groups that had the same types of people in, right? So that's all great, right? In smaller sample sizes, that sounds great, sounds very promising, but it's tough to execute on a large scale because there are no companies in Silicon Valley that are like super into having diverse leadership, not only in race and stuff, people from different backgrounds and growing up there.
I think they said that they'd rather be the fourth business to follow that model, not the first, because it is difficult to hire different types of people to give a more diverse background because it's unproven in terms of the workforce.
Well, it takes time too. You're not going to you're not going to know. It's like what's a John, right?
Yeah,
like
it's a John.
Like you're like, yo, pass me that John right there, and then you could be like, what are you talking about?
Who's John?
Yeah,
like, who's John? Who's this John fellow you keep talking about?
Yeah, so things like that. You can get used to how you describe things. Pass me that thing, pass me that John.
Yeah.
How you go about tackling problems. So there is going to be a ramp up time, right? It's just like a bell curve, though. Like eventually you're going to hit that point where, like, now that you're done kind of crawling through the stage, now you can walk and then you can hopefully run, right? But you need companies to take that chance and to try to be innovative and to try to push the boundaries in order to actually have like a real good working sample. So it's kind of like, do you wait for the data or do you just go for it?
Yeah. Well, I remember the gentleman who did math algorithms in particular for the small group analysis, where he had different algorithms that solved the problems different ways and different, maybe not even the most correct way, and then had the homogeneous group that solved it all one way, but they were super good at solving problems, and like you had said that the smaller groups that were mixed would eventually come up with the answer quicker, better, more efficiently, because they were looking at it from different points of view. I know personally, Rosa and I come from different backgrounds. I come from a split home, much poorer demographic, different things like that. So I noticed that I look at the glass a little half full.
Yep.
Or half empty rather, and she looks at it half full because she's a little bit more vibrant. She's got a better outlook, more optimistic outlook.
I think that's the easiest thing to relate it to is glass half full, glass half empty. You could be two people from the same background, from the same household even, and have a different view of things, and that can totally drive how you problem solve.
Interesting.
Yeah.
Yeah, I really did like the podcast, and we'll have to we'll throw that up in our sources as well. So it's actually about 30 minutes long. So if you actually get a chance, if you're into human resources, or if you're a you're a manager, or if you're a team builder for any type of business, I think you should check it out.
Yeah, they actually had a couple questions about. They actually had some like sample questions for people that are in HR of how to ask the type of questions to get this type of information from people. Like, how did you get here is one of the questions that they mentioned. As in, how did you get to this interview? Obviously, you're smart enough to get in this room. Tell me about some of the challenges that you've had to overcome to get here. That's a great way to kind of say, hey, tell me about what makes you different when you can't actually come out and say that as like an interview question.
Yeah, I like that. Maybe I would definitely say, I went to Temple undergrad. I had to work my butt off to get.
Dude. I mean, I had to go to Florida. I went to Florida for 4 years because I couldn't. I didn't get
a whole new way, like a whole new way of thinking by doing that. You're in a different. I know it's still in the United States, but the North versus the South, Florida versus the Northeast. That's a complete change.
I mean, bro,
well, tell me about like what type of experience that is down there that you wouldn't have necessarily up here.
Well, I'll say generally people were a lot more relaxed in Florida. Not quite the California feel, but even a little bit kind of that down south hospitality. It was pretty cool. Tampa's a pretty mixed spot, much like Philadelphia. Not nearly as large. I'd probably say maybe like a third. Yeah, maybe like 400,000 people.
Yeah, I think it's right around there. You're right.
But it's it's right by the water. It's right by the port. You got a lot of snowbirds come up. A lot of people from Minnesota, Chicago. The university has a really large international presence there, so kids from all over the place.
Interesting.
I mean, all these kids are freaking from Saudi Arabia, and all these princes driving around in their M3s. And
oh, nice! That's wild.
Yeah, it's wild,
That's cool.
Dude.
What was your? If you had an international, I know you had an international friend.
Yeah, I had a couple buddies from the UK, Matt and Davies, and Adam and Elliot and all the other guys. Yeah, I met them. That was cool. Especially when all the shit was happening with the Brexit. I was always trying to like get his opinion. Like, hey man, like, is it good? Is it bad? I don't know what this means, right? What do you think?
Interesting. Cool.
Yeah, it was cool, man. It was a good time.
All right. So I really do appreciate the podcast you got me to listen to, because I would never have dreamed of listening to a marketing podcast before. So.
Well, I mean, they tried. That one's generally like a more general internet stuff. So one of the things that one tries to do is how society is acting on the internet, how that acts, like how that affects us in like day-to-day things.
Interesting. Yeah. We all know that the Twitter. Ugh. God, Twitter.
Yeah, that can be crazy. That can be a crazy one.
But I know Twitter, they have their own problems, and I think that based upon the podcast, and especially Leslie
Miley, yeah.
Yeah, he basically said it's because they're so homogenous that if they had a more diverse background, they would have been able to get through their DM problems. They would have gotten through different things. And thought of outside the box thinking.
The whole thing right now is someone going to buy Twitter? Like, you pay attention to anything that's going on in the industry right now. Twitter's just kind of going along. They're not really doing anything super awesome. They're just kind of chugging along. So. No one really wants to buy them. They're not really doing anything breaking. Like, yeah, they're streaming NFL games on Thursdays, but no one's like jumping. Like people were like, oh, like that's cool, I guess. Like,
yeah, I heard about that. But I don't.
Yeah, it's definitely not one of the platforms that's growing anymore. So that one will be really interesting to see.
Do you think it's because of all the trolling that occurs on
Twitter?
There's
a lot of trolling on Twitter, but Reddit's full of trolls too. 4chan is literally known for trolls, and that's still a thriving community. So
I don't even know what that is.
It's another one of those really shady ones where people go and do Gamergate and go talk about all the crazy shit that they want to do on the internet.
I guess Reddit has trolls, but Twitter I feel the vitriol is so prevalent.
Yeah, well, it's because of how it's set up. It works off a hashtag. It works off of, hey, I'm going to tag something in an attempt to amplify it and have other people find it. Like, whenever I do live chats for work, so like I do my live chats and I check the hashtag, I just keep looking for the latest tweet. So people will do that with different hashtags, whether it's Bird Gang, Go Eagles, or Fuck Bradford, by the way.
Bradford, yeah, or, go Eagles, go Birds.
Or whatever it is, right? So that's how it works. So if you see someone say fuck Bradford and you like Sam Bradford, you can be like, no, he was great, he had a bad game, blah, 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 whatever you want to say.
I highly doubt there's anybody that's going to...
I'm just saying, if you're that guy. If you want to be that guy. Yeah.
But yeah, I even think people get their rocks off, so to speak, from trolling people on Twitter.
Of course. I mean, you can say it's just losers, but it's not always just losers. It's sometimes just someone who maybe they're so stressed out at work they just want to go and be shitty. I don't know.
Yeah.
It's really it's a shitty thing to do, but
Yeah. What do you think are some methods as to be able to increase the diversity of the workplace? I know in the podcast they had mentioned maybe doing a computer program to pull out African American, Asian names, white names.
That's not something that you want to do, because then you get into some ethical, like literal, like affirmative action type things. And
Okay.
you definitely want to do that. Basically, be doing the Rooney Rule, but for real world scenario, just guaranteeing the interview doesn't actually do anything.
Okay.
I think you have to just figure out ways to get people to explain that to you, to not only get in the interview room, but ask those type of questions. Not only look for kids with Ivy League degrees, but kids that have a little bit of experience. Like, we don't just look for kids that like whenever we look at interns, we don't look at kids who just come from big schools. We look at kids who have like little certifications for our field, because they're not hard to do and they're not hard to take. But if you go out of your way to take a small certification, it shows like, okay, you're going out of your way to at least try to learn more about your field. So I think those are the types of paths that we can look for, like kids that will go and will take those, whether it's trying to get into college and you go into SAT prep classes, or whether you're trying to get into grad school and you go and you do a bunch of internships or whatever it is. I mean, I think it should be more based on experience and skill than necessarily filling a quota, you know?
Fair. I like that.
Yeah.
Perfect. All right. You have
Cool.
any more points?
No, that was it. That was good, man. That was a good.
For kids who have stops. Yes. Thank you, Dave. Me too.
I agree.
Sam Bradford sucks. But all right, everybody. That was it for this week. We will upload this video from Facebook to YouTube if you guys want to check it out there.
I'm sure we can figure out how to do that.
Well, I just got to download on the phone and upload it. Yeah, that's a little easier than the simulcast. Sorry about the simulcast not working today. We had a little bit of technical difficulties. We'll have that fixed out for next week.
Yep.
Next week, we're going to do an election special. So tune in for that. Please don't troll us. And I'll say all our inherent biases at the beginning of that. And
we're going to post a bunch of sources that we talked about today in the comments after this as well.
All right, guys. We'll see you next week.
Cool. Take care.
See ya.